Welcome, everyone, to the Thermo Fisher sponsored webinar by Labroots on the topic of HRD evaluation and scoring using OncoScan CNV. My name is Mary Napier, and OncoScan is one of the products that I manage. We're thrilled to bring you two wonderful speakers today who are truly at the vanguard of understanding HRD. These innovators will talk about how they use OncoScan to assess homologous recombination deficiency, a cell's inability to repair DNA double-stranded breaks, using OncoScan. So with OncoScan, we're taking a whole genome approach, looking at the consequences. Just really quickly, I'm going to give an overview of what exactly is OncoScan. OncoScan's a chromosomal microarray that actually provides you a very high fidelity of copy number and LOH. Our assay takes about 80 nanograms of DNA, and you get from 3 days from sample to analyzed data. We have a specific platform that you see here. And these gentlemen use the output of CHAS in their methods to determine HRD. We're going to start off with Yann Christinat from the University of Geneva in Switzerland. He's going to discuss the Paola clinical trial. And then we will move on to Dr. Stephan Bartels from the Department of Molecular Pathology in Hanover's Medical School in Germany. And he's going to talk about the workflow. So both of them are very experienced. We're thrilled to have them here today. And I will now let Yann speak to his research.
Well, thank you, Mary, for the kind introduction, and welcome to everybody. So let me tell you the story about HRD in Geneva in Switzerland and how we use the OncoScan for that. So first, a short disclaimer that everything I say is actually things I say and not Thermo Fisher. And let's dig right into the topic. So precision oncology for ovarian cancer. I guess everybody has heard about precision oncology. And the whole paradigm behind it is that when you give an anti-cancer treatment, usually it's toxic. So you want to make sure that you give it to the people who will benefit the most from them. Now, ovarian cancer, we have roughly 300,000 new cases per year worldwide. And the 5-year overall survival is roughly 50%. So that's quite a deadly disease. There is some hope, though. There's a new standard of care recently, PARP inhibitors, and given as a maintenance treatment, they add roughly 2.5 years of progression-free survival, which we call PFS. Recent studies show that roughly 50% of the patient could benefit from a PARP inhibitor, but when you do the classical testing, which is BRCA mutation testing, you identify only 30%. So that's a huge population that actually is not identified with the classical BRCA test. So you could use HRD test, which is much superior, but usually it implies a high cost for a single biomarker. And in most cases, sending material into the US to a private company, which could be troublesome in Europe. Now, I told you we can use HRD. So what is HRD? HRD is a deficiency in the homologous recombination pathway. And that's the pathway that repairs the DNA double-strand breaks. This pathway is most well known because it contains the genes BRCA1 and BRCA2. Maybe you heard about the Angelina Jolie effect. And what is quite interesting is that if you have a defect in this pathway, then it leads to a characteristic phenotype, which we call genomic scars. Because if you don't have this pathway, the way the DNA is repaired is repaired in a specific manner, and we can see that in the whole genome. Now, we want to look into HRD because we know that we have a drug that is associated to that. PARP inhibitors, they work by synthetic lethality. So what is synthetic lethality? Let's assume we have a cell, and we have a table in the cell. It's quite abstract, I know. And DNA table is on the table. DNA repair is on the table. And we have one leg, which is BRCA, the other one is PARP. So all is good. Now we have a tumoral cell, which has a mutation in the BRCA. So we lose one leg. The table is unstable, which might be an advantage for the tumor, but the DNA can still be repaired. Now, if you look at a healthy cell and we add a PARP inhibitor, again, we lose one leg. It's still unstable, but it's okay. But if we have a tumoral cell with a BRCA mutation and we add a PARP inhibitor, both legs are cut down and the whole table collapses. The cell cannot repair the DNA anymore, and the cell dies and the tumor dies. So that's the whole principle. Now, the most well-known method or most used method is NGS. And the workflow, typically, you get the DNA from the FFPE sample. You need roughly 20-30% tumor content for that. And then from that DNA, you do an NGS on roughly 50,000 SNPs and some particular genes to get the mutation on, for instance, BRCA1 and 2. And once you have the copy number, then you can compute an HRD score. The most well-known is the GIS, Genomic Instability Score, which is composed of three markers, the LST, LOH, and TAI. And a sample is positive if the sum of these markers is larger or equal to 42, or if it has a BRCA mutation. Now, these three markers, roughly, they compute the same things. So the LST that we see here, if I can draw something, they compute two regions, the number of breakpoints you have between two regions of 10 megabases. The LOH, it's roughly the same thing, but only for losses. And the TAI, that's more or less the same thing, but at the end of the chromosomes. And there's been numerous clinical trials using this GIS score, the SOLO1 and the POLA1. The Athena trial used an alternative to the GIS, the GLOH score, but it turned out that this score has little predictive value. Now, the POLA1 trial is very interesting because it was one of the first trials to really clearly demonstrate the value of HRD testing for PARP inhibitors. It was composed of 800 patients treated with olaparib plus bevacizumab or placebo and bevacizumab as a maintenance treatment post-platinum therapy. And what we learned from this trial is that roughly 30% had a BRCA mutation, which is something that was kind of known. But more importantly, there was roughly 20%, which is this part here, that had no BRCA mutation, but were HRD positive. So meaning had an alteration in the homologous recombination pathway. But what is most striking is that 18%, almost 20%, one sample out of 5 had a HRD status unknown, meaning the test could not predict the response. But when it could predict the response, it was dramatic. You see here, when you have the blue line, olaparib, a nice response, much better benefit than with the placebo. In the BRCA wild type population, but HRD positive, you can still see the benefit, even though it's not as large. But in the HRD negative, there is no benefit at all. So really, there is the incentive to treat HRD positive and not only BRCA. Recently, they also published the result on the overall survival, not just the progression-free survival. And there we could see that the benefit in the BRCA mutated population, which is here, is still there. It's also still here in the BRCA wild type but HRD positive, even so it's a bit less, but as expected from the PFS. But what was striking is that in the HRD negative population, the progression-free survival results show there was no benefit, but the overall survival results show that it is actually detrimental to treat patients with olaparib when they are HRD negative. So the result is not statistically significant, but the trend is well present. Now, in Geneva, we were interested, obviously, in having an HRD test. And we could buy a commercial test, but we had actually in place everything we needed. We already had an NGS for BRCA testing, and we had the OncoScan already in place to do copy number variation. We set up the OncoScan in the lab to replace fish analysis, so that was on in 2018, and we were quite happy with it. And then when the question came, oh, could you do HRD? It's like, yeah, of course we can. We have everything we need. So the workflow is really similar to the NGS. We extract the DNA from FFPE. We need a tad less tumor content than NGS. The OncoScan is a bit more sensitive, so 15-20% tumor content. Then we get the CNVs, so it's not NGS, it's a microarray. And from the CNVs, we compute the HRD score. And for that, we don't use the GIS because there is a patent on it. We use our own algorithm, which we call the normalized LST, which I'll explain later. So the OncoScan is a SNP assay. It's not NGS, so we cannot use it to detect BRCA mutation, but it has a low cost, and we get many other biomarkers. We get copy number variation at a very high resolution across the whole genome. So we got amplification of HER2, amplification of EGFR, loss of one copy of CDK12 or BRCA1 and so on, LOH on BRCA. All of these extra biomarker, you get it for free with the OncoScan. And in our hands, it seems to be a bit more reliable than NGS, especially for samples with low quality DNA. Now, we had the test in routine use since 2020 in Geneva, and we have seen a huge increase in external requests coming from all over Switzerland. The software is available on GitHub and as our bioconductor package. So anyone who wants to play with it can download it. And that's actually what the Institut Bergonier in Bordeaux did. They got the software and they are in the process of validating it. Now, I told you we have a new biomarker. So this new biomarker, there's a rationale behind it. And the rationale is that in half of the cases in ovarian cancer, we see something that is called whole genome doubling. So a whole genome doubling happens when you have the mitosis. Instead of having, you have two copies in each cell, you duplicate, and then instead of getting two in each, one, get them all. You double the genome, and if you're counting alteration, obviously that's a confounding factor. So what we did is look at the three markers from TELIANAL, from the GIS score, in the light of the number of whole genome doubling event. And what we could see is that only the LST here retain a bimodal distribution. We want to have bimodal distribution because a biomarker should identify the two populations. They should see this camelback shape for the distribution, which we see here. But we don't see this camelback shape in the other markers. So the other markers mostly represent the fact that there is a whole genome doubling or not. So what we decided is simply to use the LST and normalize them for the number of whole genome doubling events. And what we see that when we do that, we see that the valley here is really deepening as we normalize the data. Ideally, the valley should reach to the bottom. That would be the ideal biomarker separating the two populations. There it's not ideal, but at least it seemed to be a tad better than the GIS. Okay, so we had the copy number variation data, we had the algorithm that we could test on public data, but what we were lacking is really the clinical validation, the capability of predicting the response to a PARP inhibitor. And for that, we were quite lucky to be able to participate in this ENGOT HRD European initiative. the goal of this initiative was to test on POLA1 samples different kind of HRD tests and see which one is actually feasible in routine. So there were 3 phases. The first was a meeting in 2019 to get everyone around the table, say who has what. Then there was a first technical validation on 85 samples, just to make sure everybody could do the test they pretended they were doing. And then a final validation on 384 samples. So in total, that's 469 samples. So that's not the full POLA1 cohort. And that's because we selected only DNA with high quality. But in terms of characteristics, the cohorts are very similar. And we had, in total, for the last phase, 6 laboratories participating from France, Italy, Netherlands, Belgium, Germany, and, since I'm here, Switzerland. Now, on the POLA1 trial, the HRD status was assessed with the Myriad My Choice, so we could compare our test with the NGS approach. And we could see that we had very high correlation with the Pearson's coefficient at 0.88. The positive and negative agreement value also 85 and 74. So we tend to agree more on the positive side than on the negative side. And that reflects the higher positivity rate of the Geneva HRD test, which is partly due to the fact that we have a bit more unknown sample. So all in all, what we can say is that if we use the Geneva HRD test, we get more positive samples, we have less failure rate, so more patients could actually benefit from a PARP inhibitor if you use the Geneva test. Now, what's really interesting, obviously, is the response, how the Geneva test delineates the 2 categories. And we could see that in the HRD positive with the Geneva test, we have a really nice separation between patients that receive olaparib and placebo for the progression-free survival. And the hazard ratio is really similar to the NGS approach at 0.41. On the HRD negative population, we see the same result as on the POLA1 trial, no difference between the 2 populations, depending on if you get olaparib or the placebo. Now, what is really interesting is that on the overall survival, we again see, as Isabel Recocar showed on the POLA1 data, a benefit when it's HRD positive. But when it's HRD negative, which is here, we see that the effect is really more drastic than the data showed at the last ESGO. And it's really detrimental in the HRD negative population to give olaparib in the long term. There's no benefit of PFS, but in the long term on overall survival, it has a worse effect. And with the Geneva test, the results are statistically significant. Now, obviously, we have 2 tests. We want to look at the differences. And in the BRCA wild-type subpopulation, we identified 28 samples in green here, which were negative for the NGS approach and positive with the Geneva test. And when we look at these samples, we see that they tend to follow the blue line here. So they respond well initially, but then many of them recur more drastically than the blue line, which is the Geneva positive and NGS positive. So this is really a subpopulation that is quite interesting. Unfortunately, we don't have that many samples to actually make anything statistically significant, but that's something we're really willing to investigate a bit further. So that brings me to the next development. Within the ENGOT initiative, we have the result from the 6 different tests, the 6 different labs, and there's going to be an analysis to focus on discordant samples to see what are the differences between them, which could lead to an improved test in the end. And on our side, we're going to collaborate with Dr. Labidi Gali, who recently showed on the whole POLA1 data that depending on the location of the BRCA mutation, it has an impact on the PARP inhibitor response. So we want to combine these 2 information with this partial phenotype that we've seen here and the information about the mutation to make sure that we have the best available test for our patient in Geneva. And for that, we need to analyze the rest of the cohort. of the POLA1 cohort. So that's roughly 300 patients. And what is interesting is that these 300 patients, they're going to be a much lower quality because all the high quality samples that were selected for the ENGOT initiative. So that's going to give us a better proxy to a real life situation in terms of test failure. Now to conclude, we've shown a really high concordance between the NGS approach and the Geneva HRD test, a lower failure rate for the Geneva test, and also the software is open source, so anyone who wants to implement it in his lab, it's available online. And last but not least, all of these results together suggest that more advanced ovarian cancer patients might have the opportunity to benefit from a PARP inhibitor if tested with the Geneva HRD test. With that, I'd like to thank you for your attention. I'd like to thank the lab, the POLA1 and HRD teams, and AstraZeneca. And if you have any question, that's going to come later. Now, I'm going to leave it to Stephan, which is going to talk about his experience with OncoScan data. Stephan, the floor is yours.
Thank you, Yann, and thank you, Mary, for the kind introduction in the beginning. Thank you, Thermo Fisher, for the invitation. I'm Stephan Bartels, a PhD researcher at the Hanover Medical School in the molecular pathology lab in the Institute of Pathology. And I'm responsible also as vice head of the lab for the establishment and validation of new lab techniques. And I want to share with you our results of more than 2 years HRD testing with the OncoScan assay and the results from our clinical research, the performance with the assay and the pitfalls. Sorry. So this is my disclaimer, same as Jann's. These are the contents I want to talk a bit about with you. The technical background and the lab workflow in the beginning. Then I want to show you our clinical research samples, research projects we performed in the last years with the OncoScan platform, where we investigating other topics and advantages and some disadvantages of the OncoScan platform regarding HRD testing and future developments. as Yann already showed. So up to 40%, close to 40% HRD when in ovarian cancer when BRCA1 and 2 is wild type. Okay. We are also part of the German HRD consortium. This is an academic consortium which want to test HRD with various methods, laboratory methods. 7 German university hospitals are participating, and we use 6 molecular HRD assays. All assay is tested by at least 2 different labs. And we had FFPE DNA samples from clinical studies with known GI scores. And the tests are already analyzed, everything is finished, and the manuscript is written, and hopefully will be published soon. It is in revision at the moment, and we got quite friendly reviewer comments. So I hope that in the next week we will have the acceptance of this study. So, and these are one of our research projects from the last years. This is a breast cancer case with 2 regions, 2 tumor regions in the breast with a complete different morphology. And at first we thought that this must be 2 different carcinomas with completely different origin. But then we do comprehensive molecular analysis, and these 2 different regions share the same CDH1 frameshift mutation and share different amplifications and deletions analyzed by the OncoScan. They share most of them, and some are individual. So we conclude that these 2 tumor regions must have the same ancestor lesion and underwent a tumor evolution in follow-up. So this is another study from our lab in cooperation with our human genetic department. There we analyze a cohort of myoepithelial-like metaplastic breast cancer cases, and the ancestor lesion of these carcinomas. And we could show that these metaplastic breast cancers share PA3 kinase mutations and a loss in chromosome 9 on the long arm. Maybe you can see this thin blue line here in most of the samples. It's the region of P16 CDKN2A and P15 CDKN2B, and this region is deleted in most of these samples. And this is a quite well diagnostic biomarker for these carcinomas. And chromosome 5p gain is important for the prognosis of these cases. So advantages for the OncoScan array platform is that the molecular inversion probe technology is developed for FFPE material. Therefore, it is a very robust workflow. You have an easy data analysis. You can use very low, very limited DNA input amounts. You can perform copy number analysis also for other tumor entities. And it is also possible to perform gene expression experiments on the array platform for research projects too. The disadvantages, you cannot get the information about mutations. It's not really true because the OncoScan assay, one version of the OncoScan assay can analyze hotspot mutations in addition, but it's only hotspot mutations. So that's not such a big amount of information. And in Germany, I'm surprised that Yann says it's cheap, because in Germany, the costs for one OncoScan analysis are higher than for the comprehensive NGS panels, for example, OncoMine Comprehensive Plus. So, but maybe that's different depending on the region your lab is located. Okay, some data, what will come up in the future. We have studies for PARP inhibition therapy in urothelial cancer, for example, the Atlantis phase 2 trial. And there we see a very nice increase in progression-free survival in patients with deficient DNA damage response. We have studies before, and they compare unselected urothelial cancer patients for PARP inhibitor therapy, and the results were globally negative. So these authors concludes that the prior selection of the HRD status is essential for these patients to benefit from the PARP inhibitor therapy, and hopefully we can test them in the future. Melanoma patients have approximately 10% showing HRD, but we have only limited studies so far. Some case reports show a partial response. We have to see. And prostate cancer prostate cancer patients have an increased progression-free survival when they are BRCA1 or 2 or ATM mutated, but not that much. And the effect is even lower when you look at other HRD mutations. Then it is very, very, very small effect. So my conclusions are: OncoScan is a very robust lab technique, easy to implement. The data analysis is easy to implement in the lab. The FFPE tumor samples can be analyzed, even small tissue samples with low DNA concentrations and low tumor cell count. And most importantly, approximately 40% of BRCA1 and 2 wild-type cases, ovarian cancer wild-type cases, are HRD and could potentially profit from PARP inhibitor therapy. Yann already showed the results from the Paola trial. And in the future, further tumor entities are possibly treatable with PARP inhibitor therapy. So thank you for your attention, and I'm looking forward to the questions. Thank you.
Thank you, Yann and Stephan. I'm going to welcome all three of us back on camera for the questions. And I see that we have several questions coming into the queue right now. In the event that your question is not answered today, please know that your question is important, and we will work towards answering each one and then communicating with you about the insight from your question. Okay. So this first question here is for both of you from your perspectives. So we heard Dr. Bartels or Stephan talk about several types of samples that he evaluates for HRD. So it feels like we look at more than just ovarian samples. Can both of you give a feeling for what tissue types you're looking at is in addition to ovarian?
Yeah, maybe I can start. As I said, we already analyzed breast and endometrial cancer on a regular basis, so not that much than ovarian cancer. And so the most promising candidate for me is the urothelial cancer at the moment. So I'm very optimistic that we will get samples in the future.
As for Geneva, we so obviously the main focus is on ovarian cancer, but we have what we call this molecular tumor board, where it's a tumor board where we get all kind of samples, people that have run out of treatment, and for these samples we're going to do the OncoScan, because if we see an HRD positive, maybe that's an indication for a response to a platinum therapy or something like this, or PARP inhibitor. So in this indication, we're going to do the OncoScan on whatever arrives here.
Yeah, correct. So for example, pancreatic carcinomas, we already performed this a few times because pancreatic carcinomas are also BRCA1 and 2 mutated sometimes, and yeah, in the molecular tumor board, I think it's the same in your hospital. Patients in the end stage where we try everything.
Yeah, we just emphasize the fact that the threshold that we use of 15 is validated for ovarian cancer. If we have a sample like a pancreatic cancer, which an HRD of 16 says, well, always be cautious. It's not the HRD test has not been tested for all the cancer type. So
that's true. Yeah.
I love that. Always be cautious. Okay, so here is another question for both of you that both of you actually touched upon. So we know that HRD is detected by the causes, the mutations in the repair pathway, most notably BRCA, as well as several others, and the consequence is genomic damage. So OncoScan, great at looking at genome-wide damage. But how are your laboratories determining the BRCA status?
Maybe I'll start this time, Stephan, if you don't mind. So what we do is we do an NGS panel to assess BRCA1 and BRCA2 mutation. So we don't do the methylation, but the main idea is that actually the HD score is superior to the BRCA mutation status. This is what we believe. And the BRCA mutation status really help us salvage, because it's a bit more sensitive in terms of tumor content. So if we have a sample with very low tumor content and we cannot run the OncoScan because it has, let's say, 10% tumor content, the NGS will detect the BRCA mutation at 5%. So those cases can be rescued. And that's the high value of making NGS and detecting BRCA mutation.
Yeah, also in Hanover, we have a few NGS panels which are covered BRCA1 and 2, focused smaller NGS panels or more comprehensive NGS panels depending on the tumor entity. We analyze also the BRCA1 hypermethylation also with NGS.
Interesting. I love the incorporation of methylation. I think that adds an interesting dimension. Okay, on to the third question. Yann, your group finds this particular person has asked, your group finds the combination of OncoScan and NGS as the best approach. But what are the benefits of using OncoScan over NGS alone for HRD detection? And if you, Stephan, want to add in, since it sounds like you also are using an NGS OncoScan, that would be helpful as well. But let's first start with you, Yann.
Yeah, so the benefits that we have from using OncoScan, one is a very simple reason. The NGS panel is already there and the OncoScan is already there. And if we need to switch from modifying the NGS panel, that means validating a new assay, and this is complicated. But besides that, there are really other good reasons to prefer the OncoScan over the NGS, is that as I said during the talk, we find that the OncoScan is a bit more robust in terms of DNA quality than the NGS. And also, we managed to get a lower tumor percent. The requirement for the tumor percent is a bit lower with the OncoScan than with the NGS. So that's the main.
Yeah, correct. Yeah.
Laziness and.
Dr. Bartels, it sounds like you agree. Anything else you want to add, Dr. Bartels?
I agree.
Dr. Bartels,
yeah, and to add one argument, it's for me it's always important what the technique is built for, and the genomic array technology is built for detecting amplifications and deletions, and NGS is built for detecting mutations. And for sure, you can calculate copy number variations from NGS data, but it's not built for that. And I think that's the reason. We also see that the OncoScan, like Yann, that the OncoScan is more robust when you have samples, especially when you have samples with lower quality, DNA quality and DNA amount. And I think that's the reason. And the GI score is more precise, for my opinion, because more SNPs are analyzed. And even in a large NGS panel, you analyze less SNPs than in the OncoScan array. So the calculation cannot be so precise.
Our design backbone is very dense vis-a-vis hybrid capture approach. Okay, let's see if we've got another.
I think to add on that, I think that those two are really complementary, because with the OncoScan you get very high resolution on the genome-wide, which you cannot get with the NGS. But on the NGS you can get like exon-specific deletion on specific genes. So using the two, I guess you get the best of the two.
Just waiting for a couple of questions to populate here. Okay. Reading questions. Okay, Dr. Bartels, these questions are for you, and they're all kind of related to non-optimal sample quality. So can you explain this from your perspective? I think, do they end up in inconclusive results? What are some of the thresholds you use to evaluate if something will work on, let's say, OncoScan?
They are mostly not end in inconclusive results. We have a few sample dropouts, less than 5%, but we see a lot of more samples with non-optimal DNA quality. This is a bit special for our lab, I think, because we get external samples from all of northern Germany. Many of them from small hospitals, small pathology labs, and often they do not have the best protocols to fix the tissue and to pre-process the tissue, the FFP, and yeah, that's a big advantage of the OncoScan assay, that we have so many samples we can analyze despite of this bad quality. So normally, we try to get good results for sure, yeah. I'm feeling more comfortable when I have an HRP result from a bad quality sample, because it's not, the risk is not really high that you have a false negative or false negative HRP result, but to get a false positive HRD result from a bad quality sample, that's quite high. And yeah, therefore you can optimize your data analysis platforms, repeat the sample, ask for a different tumor block or different biopsy material if you can get some. And but yeah, for most of the samples, it worked quite well.
Excellent. Well, is there anything that we haven't asked today that you might want to add before we close?
Not from my side.
Excellent. Great. Well, thank you very much for everyone who's attended. We were very fortunate to have such rich materials over clinical trials that clearly took a lot of dedication over years to conclude. We want to thank Labroots, thank Thermo Fisher, and our speakers. Everyone have a wonderful day.
Thank you all. Thank you.
Thank you.